transport chain, and we're illustrating pretty much what we just said so far. So let's look at where we are kind of zooming in on. So here's our mitochondria. Here is the inner mitochondrial membrane. Remember, the mitochondria is this double-walled organelle. And so what we're zooming in on is this layer here. So we're taking a closer look at both mitochondrial membranes. So here's the outer membrane, here's the inner membrane. This region in here is called the matrix. So let me just point that out here as well. This is the mitochondrial matrix. And then this is the intermembrane space. I'll just put IMS here, the intermembrane space. So basically we've got the outer mitochondrial membrane here. We've got the inner mitochondrial membrane here. Oops. Okay, so let's label these out. Outer mitochondrial membrane. Versus the inner mitochondrial membrane. Oops. Okay, and so we describe this region as being the intermembrane space. Alrighty. And so the first thing we want to put in here in our drawing are these complexes that are embedded in the inner mitochondrial membrane. So we've got a series of protein complexes that are embedded in our mitochondrial membrane, the inner mitochondrial membrane. And then the final complex, I'm going to draw it in green here, is probably the most important one for this process. This is called ATP synthase. Okay. So this is ATP synthase. Oops. We also want to establish that there is a gradient in between the inner membrane space and the matrix. So remember, down here is the matrix. Let's label this here somewhere. So here's the matrix, which is this central region. So just to bear that in mind. And so across this space, we have this gradient set up where there is a higher concentration of electrons out in the space here, or hydrogen ions rather. So we can say hydrogen ions, electrons, pretty much synonymous. Charged particles out here. So we have a higher concentration here. And we have a lower concentration out here in the matrix. So very few present down here in the matrix. And so what we have set up is this gradient already. Let's talk about how we actually achieve this gradient. This is where our electrons come into play. So all of those electrons that we've been garnering thus far, these are brought into the matrix and they are passed from complex to complex. So we take our electron carriers, pretty much NADH, oops, NAD, H, remember this is in the reduced form carrying electrons. We also had another carrier, right? We had FADH as well. 2, because it's also carrying electrons. And so these are our carriers coming in from our other processes, the Krebs cycle, glycolysis, and our linking step. And they automatically get oxidized where they basically shuttle those electrons from these carriers onto complex 1. So what happens here is we oxidize these carriers. Remember, oxidation is loss of electrons. So we kind of just pass these electrons from these carriers to the complex. And so on the other end, we end up back with the oxidized form of our carriers, which are NAD plus and FAD. empty, no longer carrying electrons, okay, just to make this process clear. And so now the electrons are at complex 1. As they go through complex 1, they move from this, the carriers here, through this complex, right? Let's just make that clear here. And so as these electrons pass through complex 1, we then have the passing of this energy from complex 1 to complex 2, okay? Likewise, as we pass electrons from this complex to this complex, we pump some more hydrogen ion across this membrane as well. Okay, we once again pass electrons from complex 2 to complex 3, and as we do that, we generate the energy to pump some more electrons across the membrane. So can we see how the electrons are creating the energy that allows these complexes or these pumps to pump these hydrogen ions from the matrix over into the intermembrane space? And so this is how we establish that gradient that we now have going on. We have more hydrogen ions present on this side of the membrane, less hydrogen ions present on this side of the membrane because of the energy that's liberated as we pass these electrons that are coming in from one complex to the other. And I've got these complexes here drawn in red just to highlight that this is an active process. Active because it's going to require energy to move these hydrogen ions across these complexes. And so as we're moving these hydrogen ions, through these complexes from the matrix out here into the intermembrane space, we're taking the energy that's coming in from passing of these electrons. Okay, I hope that's clear. Now, what we have at the end here is oxygen standing by to be the final electron acceptor. So we have oxygen here. This is oxygen. And so the very last complex can pass its electrons onto oxygen, and this actually creates water. Okay, this is the whole sole purpose of us having oxygen in our cells, to be the final electron acceptor, to keep this chain moving, to keep the passage or passing of electrons from one complex to the other. And because of the gradient that we have now set up, right? So we talked about this gradient, we talked about how we get the gradient, how we achieve the gradient, right? Really through the passing of electrons and the pumping of these hydrogen ions. But the importance of the gradient is that we can now pass all of these hydrogen ions through the final complex here, which is ATP synthase. And I'm drawing this here in green to show that this is a passive process, and this is going to liberate the energy that actually phosphorylates ADP and makes ATP. So now we have a gradient here. The high presence of hydrogen ions now can move right through ATP synthase, and this is what allows for the energy that actually phosphorylates ADP and makes ATP, okay? So I'm going to draw that here. As these hydrogen ions move through ATP synthase, we now go from ADP plus the phosphate, and we make ATP, okay? Now, because this is oxidative phosphorylation, this is the electron transport chain, we make the bulk of our 38 ADP. We make about 34 ATP molecules per molecule of glucose.